Hi everyone, this is Tony Yap, a master student of genome biology in University of Malaysia. In this short video, I'm going to talk about whole genome sequencing. Whole genome sequencing has been a subject of hype since the first human genome project started, but unfortunately, the general public has very little understanding of this topic. So I'm going to explain this concept fairly easily and simple, so simple that even a 3 years old kid is going to understand it. The purpose of doing whole genome sequencing is to acquire the entire genetic blueprint of an organism so that it could be decoded and be read like a book, literally reading a biography book which tells you everything about the author. In this case, it tells you everything about the trait and characteristics of an organism. The understanding of the organism has immense value to the society, which ranging from great scientific breakthroughs to saving millions of lives. As we learned in basic biology class, the genetic code is made of DNA, which comprises of 4 chemical letters, namely A, T, C, and G, which collectively is called the bases. It's how we string these bases together to make a trait, pretty much like how we put different words together to form a meaningful sentence in the human language. But there's a problem with these letters. They're not readable. Unlike the font size 12 of the Times New Roman that we use daily, this letter is extremely small, so small that even the world's most advanced microscope fails to read them. But fortunately, we have this machine called a sequencer which could perform the task. During the genome sequencing process, the DNA is first chemically tagged so that it could emit signal and be detected by the sequencer. But there is an inherent problem to this as the genome is extremely large. You imagine if you were to read a thick encyclopedia books from end to end, it could easily take you years if not decades. So a better approach to do this is by a shotgun strategy, which means that to break down the book into many pieces or pages so that it could be read simultaneously in a parallel way. After the sequencers will generate whole lots of data in the form of reads, which ranging from millions of bases in bacteria to billions of bases in the case of complex organisms like humans. As we learned earlier, these bases are in the form of fragments which have to be pieced together to form the complete genome as we do for puzzles, and this cannot be done manually. Doing manually is no trivial and no man could ever do this. If you were to do this manually, you would either go crazy or you would quit science entirely. A smarter way to do this is by feeding them into the computer system, in which the algorithm will run a series of repetitive action to align and sort them together to form the complete genomes. And now you have the complete genome and you can do all kinds of downstream applications to answer your research questions. It's just as simple as that. Thank you.